And I want to welcome everybody and say good morning or good afternoon or good evening, depending on where you're calling in from. And welcome you here to ICCVAM's 2025 Annual Community of Practice. My name is Helena Hochberg, and I am the Acting Director of ICCVAM, which is the U.S. federal resource for alternatives to animal testing. ICCVAM provides scientific and operational support to ICCVAM, which is an interagency committee composed of representatives from 18 federal regulatory and research agencies that all require, use, generate, or disseminate toxicological and safety testing information. So ICCVAM and ICCVAM promotes the development and validation of toxicity testing methods that protect human health and environment.
Your slide is not showing, Helena.
No, I'm not having a slide. I'm not having a slide.
Okay.
While replacing, reducing, and refining animal use. So ICCVAM also provides guidance to test methods developers and facilitates collaborations that promote the development of new test methods. And to address these goals, ICCVAM holds an annual Community of Practice webinar. And this year, we're going to focus on evaluation of complex in vitro models, which is very close to my heart. That's how I started my research here in the United States. It's a very timely topic, considering the recent press releases from various agencies, especially from the FDA. And we have three really excellent speakers lined up for you today that will take us from the development of these models through qualification and validation, and finally into applications. So we will finalize with a combined Q&A to facilitate discussion. So we'll start with the three presenters, and then there will be a combined Q&A. So there's some logistics about that. The chat is disabled for you, but please use the Q&A to ask your questions. And if your questions is for a specific speaker, add the name of that speaker, please. You have the option to upvote questions, so we will take those first. So if you like a question, please put a thumbs up. And if you don't see your questions anymore, you can look in the answered section because there might be a written answer to it, or we might answer it live. A recording and the slides will be available on ICCVAM's webpage once it's formatted. And with that, I want to go ahead and introduce our first speaker, which is Dr. Dan Tagle. And he is the director of the Office of Special Initiatives at NCATS NIH, which is actually our most recent formal ICCVAM member. So they joined us last year formally. In his role, he leads strategic programs to accelerate translational research and innovation across biomedical fields, where he played a key role in pioneering technologies such as microphysiological systems, or MPS, 3D bioprinting, and synthetic biology to advance drug and diagnostics development. He also leads a team in developing quantum technologies, electronic nose technologies, and automated chemistry integrated with AI and machine learning. So with that, I'm not keeping you any longer. Let's go ahead and hear Dr. Tagle's presentation.
Thank you very much. Good morning. Thank you, Elena, for the kind introduction, and certainly to the ICCVAM team for facilitating this webinar. So good morning to everyone. So I'm here to give you a big overview of complex in vitro models, otherwise known as microphysiological systems, tissues, or organs on chips. But let me start with defining the problem for us by telling you in terms of why NCATS exists and why we're trying to develop new technologies. So over the past couple of decades or so, certainly we as a group of scientists have become better in identifying diseases through genomics, through the Human Genome Project, and various omic technologies, to the point that we can now identify about 10,000 diseases. However, this understanding of the causative genetic causes of disease have translated poorly into bona fide treatments and cures. So in fact, only about 5% of those 10,000 diseases have identified treatments or cures. And for the most part, what we're trying to address at NCATS is that it has a very high attrition rate. As you see in the top, there's 9 out of 10 candidate therapeutics fail in clinical trials, and it often takes 10 to 15 years at a cost of $2.6 billion to move a candidate drug into the clinical trials. And quite often, if you look at across very many indications, as you can see here, the 9.6% is the average success rate. And while we do well in things like hematology and infectious diseases, doing well meaning around a quarter of percent success, we do fairly poor in psychiatry and oncology, which is a very low success rate. And part of that is that the known animal models do not show the same level of concordance of toxicities within humans. And so what we really need are human relevant model systems that can reliably predict health outcomes that often can vary because of sex, age, genetics, of course, population variability, and also individual differences. So what I'll show you next is a video telling you what microphysiological systems are all about.
Several thousand diseases affect humans, of which only about 500 have any treatment. Getting a drug through the current development pipeline takes too long, is too costly, and the vast majority of new therapies, about 95%, never make it past clinical trials. Drugs that seemed promising in early studies using cell and animal models often don't have the same response in humans. We need an alternate research model that accurately represents the human response. NCATS, the National Center for Advancing Translational Sciences at the National Institutes of Health, is supporting research to make this possible. Through the Tissue Chip for Drug Screening Program, a collaboration among NIH, DARPA, FDA, and researchers nationwide, NCATS is supporting research on bioengineered devices that mimic the function of human organs. The goal is to use these tissue chips to test the safety and effectiveness of potential new drugs, weed out those that don't work, and pursue ones with the most promise. The chips are fabricated from a flexible plastic with ports and channels to allow nutrients and oxygen to pass through them. Unlike cell culture and animal models, the innovative design of tissue chips replicates the complexity of the human body environment. One example is a liver chip, which includes multiple cell types that are organized much in the same way as they are in the human liver. Features such as blood flow can be mimicked to recreate what cells experience in the body. The complexity of these chips generates human-specific data, which enables researchers to better understand how the liver responds to a drug and if it is toxic, critical data before testing in people. By connecting chips that represent various organs into a full human body system, scientists may better understand how the whole body responds to drugs and if they're safe and effective. For example, chips that include additional factors such as hormones could help researchers investigate differences between male and female responses to drugs. The ultimate goal is to use these chips in a variety of ways, including to advance precision medicine, test for environmental toxins, and develop disease-specific models such as for cancer and rare diseases. By developing, demonstrating, and disseminating tissue chip technology, NCATS is improving health through smarter science.
So just to give you then a sort of like a wider overview of what's been going on in the past decade or so, we have NCATS have supported programs in safety pharmacology, That was in the initial start of the program, and then went on to see if we can actually capture models of diseases in these organs on chips, and thereby use them for efficacy testing. And then lately, we just concluded a program on clinical trials on chips for the study of precision medicine, and then building confidence as well as building community in terms of adoption of the technology, and then lastly, regulatory acceptance. I'll be touching a little bit on some of this through some presentation of some case studies, but just wanted to point out that this is largely participated in by many institutes and centers at the NIH, many government agencies such as the FDA, as already mentioned, DARPA, NASA, BARDA, Vitro, the Veterans Affairs, EPA, as well as working with various pharmaceutical companies. So the question then is, are tissue chips really better predictors of human response? And so let's just take an example of the liver, which is primarily the organ that's responsible for concentrating and metabolizing a number of medications. And because of that, drug-induced liver injury, of course, is the most common cause of acute liver failure, and can cause hospitalization and liver failure that can result in transplantation. So typically, a human liver on a chip would consist of the hepatocytes, along with non-parenchymal cells such as stellate cells, Kupffer cells, and endothelial cells, and will have perfusion or flow into it. And so in one of the studies, and this is a study done by the Emulate, looking at a response of a drug called phenyluridine. Phenyluridine is an antiviral agent that was developed for hepatitis B. When it went through preclinical testing in rats and dogs, it went through to the point that FDA approved them for human studies. But lo and behold, it actually killed 5 out of the 15 patients that were enrolled in the first in-human study. And so to understand whether human liver on a chip can actually predict or anticipate the toxicities that the rat and dog model was not able to predict, rat and human hepatocytes were put on the chip and dosed at different levels of phenyluridine from 0 to 30 micromolars, and stained with Nile red. Nile red, it's a stain that will a stain for lipid accumulation that would indicate steatosis. And so you can see here in the micrograph, it shows that at varying doses, there's a dose-dependent increase in toxicity or formation of lipid accumulation and steatosis. And that could be quantified on the right-hand side on the graph, showing again the dose-dependent toxicity. One can also look at the functional response. And so if you look at albumin, which is a common functional secretion of the liver, you can see that the rat hepatocyte on the chip still continues to secrete albumin even at a high dose. Whereas in the human hepatocyte on the chip, you can see that there is a dose-dependent decrease also in albumin secretion. Likewise, you can look at markers for liver injury, such as miR-122, glutathione S-transferase, and keratin, and show that there is a dose-dependent increase in the expression of these liver injury markers. One can also then extend this study. And in this case, if you look at the table, those additional 22 compounds that were tested. These are just like the phenyluridine was tested in preclinical models, went into clinical trials, but unfortunately, in the conduct of the clinical trials, killed 200 patients and caused 10 liver transplants. And so clearly, the safety signal was not anticipated in the preclinical models. And so using a human liver in a chip, just like the testing with the phenyluridine, the human liver in a chip showed 87% sensitivity and 100% specificity in predicting the hepatotoxicity that was not predicted in this platform. Another study to point out would be in terms of efficacy. So this is using a tissue chip model to model an autoimmune demyelinating neuropathy. In this case, the tissue chip consists of human primary Schwann cells, cultured with iPS-derived motor neurons. And since this is an autoimmune disease, part of the study is taking anti-sera from patients. So in this case, each of the panel here consists of one patient. So there's 15 anti-sera, all staining for complement activation in these tissue chips, as shown by staining for C3b, shown in green, and CBB9, shown in red. Then if you take a candidate therapeutic that was developed by Sanofi, in this case, it was a monoclonal antibody that inhibits a C1 protease, and pre-treat the neuromuscular junction on a chip, you can see that the complement deposition on these 15 patient chips did not elicit the immune response. And so they showed it was protective and showed efficacy. And so this data was then used to support, which is now currently a phase 2 clinical trial for this candidate therapeutic for neuropathy. The other application that we've been doing that we just concluded was the clinical trials on a chip. And so this is actually using the platform to populate the chips with various patients, representing essentially the patient demographics, whether it be sex, age, gender, and also genetic mutation. What we hope to accomplish here is to establish a recruitment criteria by which to improve clinical trial design, be able to stratify the patient population in terms of those who are going to be the best responders versus those who are likely to develop adverse events, and then also to develop clinically relevant biomarkers. And so one study is in NAFLD, the non-alcoholic fatty liver disease. This is a study done at University of Pittsburgh with Lance Taylor as a PI. And it consists of taking various iPS-derived, in this case, 100 subjects, 50 males, 50 females, directing them to differentiate into hepatocytes and non-parenchymal cells, and then being able to form this vascularized liver acinus model, or VLAMs, and then testing for the clinical efficacy of obeticholic acid as well as pioglitazone, and comparing that to metformin and ursodeoxycholic acid that have already been known to fail in human trials. And so this is an example where we can look at a very specific, in this case, a very specific mutation, the PNPLA3 gene that is very common in the Hispanic population, and indicating that they are in some ways very high risk for developing this non-alcoholic fatty liver disease. Another area to highlight would be how we build confidence into this technology, since we started it about a decade or so ago. And so, as I mentioned, a number of projects were funded to demonstrate not only toxicity and safety testing, but also efficacy. And so in that context, demonstrating physiological relevance of the tissue chips, and getting those into the literature. So as of October, just for this cohort of projects that were funded, more than 500 publications that have been out in the literature. And then back in 2017, what we wanted to do was also to do analytical validation. That is to test for the robustness, reproducibility, and reliability and relevance of the tissue chips. And this is done through independent testing centers. A couple of the testing centers that were funded by NCATS was at MIT and Texas A&M, as well as creating a database that would share the data related to tissue chips. And then we encourage these testing centers to become a self-sustaining business model. And so the MIT center developed into a CRO business model, and is now Javelin Biotech. And you will hear in the next talk that the testing center from Texas A&M became the TexVal Consortium, which is essentially a pay-per-play model working with academia, government, and industry. And then subsequently, the MPS database also is now a business model that is operated by Curious AI, and it's called Eve Analytics. And you'll hear more from Yvonne Rusin about the TexVal Consortium at the next talk. One of the things that we also did was to move tissue chips in terms of regulatory acceptance as qualification as drug development tools. And so we work with patient groups, the pharma companies, regulators, patient advocacy groups, academia, government agencies, essentially to establish centers and to enable MPS or tissue chips to advance them as drug development tools, and essentially to qualify them in terms of a specific context of use. So this is done with active partnership with the FDA and the Critical Path Institute. And ultimately, the end result would be to enable assessment of drugs for the right target, the right person at the right time, and be able to show and demonstrate that this platforms can be used effectively as drug development tools. And so in that context, as we have a letter of agreement between NCATS and the FDA that also involves the Critical Path Institute, and these centers are tasked with developing MPS that are fit for purpose for industry needs and have specific context of uses. And as shown here, there are 4 centers currently funded, one at Mount Sinai University, as well as University of Washington, that is focused on a group of MPS platforms working on kidney on a chip. And then there's the University of Texas Medical Branch at Galveston, Texas Engineering Station in Texas A&M, headed by Ramkumar Menon. And this is focused on preterm birth and female reproductive health. And then there's the Liver Center at University of Pittsburgh, headed by Lance Taylor, and University of Rochester and Duke University, headed by Jim McGrath, focused on barrier function. And so these centers are actually in the process now of working in terms of submitting letters of intent to the FDA drug qualification process for drug development tools. And happy to note that even in the early stages of funding, we just funded this centers just beginning of last year. They were already able to successfully submit in several instances letters of intent, and in a few cases actually have accepted letters of intent. Another highlight to show is that not only can tissue chips be used here on Earth to advance science, but we can also develop them for use in space research, not only to study the effects of chemical exposures in space, as well as biological hazards and environmental hazards, but also to be able to anticipate what future space colonizers would experience as we move towards Moon and Mars. But more importantly, at NCATS, we wanted to understand how the effects of the space environment can actually lead to advances in translational medicine for use here on Earth. And so the classic study on that is the twin study, where in this case, Mark Kelly, shown here on the left, and Scott Kelly, which actually went to mission and spent more than a year in space. And shown here are the various changes from their immune system to the metabolomics and microbiome and various other changes that happens to the human body, mostly as an effect of microgravity and in some ways mimicking the effects of accelerated aging. And so back in 2017, NCATS partnered with NASA and the Center for Advancement of Science in Space, as well as being able to send various payloads through SpaceX to study immunosenescence, the effect of lung infection and bone marrow, mobilization of the innate immune response, the formation of kidney stone, osteoarthritis, changes in blood-brain barrier permeability, cardiomyopathy, also gut inflammation, and being able to study how sarcopenia or muscle wasting can also result in space. But more importantly is being able to translate those findings for use here on Earth. And then I'd like to play for you a video from the astronauts who've actually worked on these tissue chips in space.
Greetings from the International Space Station. I'm NASA astronaut Jasmine Ogbeli, along with my crewmate, Dr. Satoshi Furukawa of the Japan Aerospace Exploration Agency. My crewmates and I have been hard at work supporting an array of research experiments and technology demonstrations in this microgravity environment. The space station is a unique laboratory, unlike any other, where there is no gravity. And because of that, we can look at science in a new way to benefit humanity.
One of the areas of tremendous interest is research in human cells and tissues through a lab on a chip, or what we like to call tissue chips. We know that the space environment has profound impacts on human physiology, and through tissue chips, we can seek new avenues for drug discovery, potential therapeutic development in the space environment.
I encourage those in attendance to think about taking your research beyond Earth's horizon. Through space-based research, we can hopefully find ways to treat many of the mitigating ailments that humans develop on our planet. Thank you for all that you do to treat patients on Earth.
Yeah, and so we're also in the process now of renewing this program. It will be announcing the funding of several new projects that will go up into space in the near future. One of the exciting things that's been happening lately too is the area of NAMs, or new approach methodologies. And so this is in some ways taking into consideration other model systems, not just in vitro, which is what MPS or tissue chips are, but being able to combine them with in silico or computational modeling as well as in chemical. And so one can think about complex in vitro systems being integrated with in silico and in chemical, and being able to combine them into a much more comprehensive combinatorial NAMs with a common data ecosystem. And so this new program is actually in the process of being stood up at the NIH. It's a program called Complementary, for complement to animal research and experimentation. And we're in the process of identifying technology development centers, as well as a data hub to support NAMS development, and also a public-private partnership called the Validation and Qualification Network that would work with various regulatory agencies as well as private partners to support the validation and qualification of these new approach methodologies. And then I'd like to also point out that this market, the global market for tissue chips, is actually growing. It's projected to reach $1.6 billion by 2030 with an annual growth rate of 35.1%. So there's active areas, and you will hear from Ivan Rosenstock about many of the small businesses, either startups or spin-off companies that have formed in this area. Helena, in her introductory remarks, talked about also the excitement in the field because of the recent announcements from various agencies, starting off with FDA announcing back in beginning of April a roadmap to phase out animal testing and focusing on the use of new approach methodologies, especially in the testing of monoclonal antibodies and other small molecules and biologics. And at the same time, actually at the same day in the afternoon, the EPA Commissioner Lee Zeldin also in some ways recommitted to their roadmap on a ban on animal testing at the EPA. And then towards the end of April, the NIH also put out a press release indicating that the NIH is prioritizing human-based research technologies. And then at the beginning of June, the NIH and FDA also announced a new joint venture that will focus on nutrition, and then primarily looking at microphysiological systems for chemical risk assessments for food additives and certain food chemicals and preservatives, being able to harness these new emerging technologies and how we can better develop better food products for our community. Lastly, in terms of building community, NCATS have been supporting an annual World Microphysiological World Summit. Started back in 2022. in New Orleans that was attended by close to 500 attendees. And then we went on to in 2023 in Berlin. It alternates within United States and Europe. And then Seattle, Washington last year. 2025, this year was in Brussels, Belgium, and was attended by close to 1,500 attendees. And happy to note that next year it will be May 26th to the 29th here in Washington, D.C. at the Washington Convention Center. And so we're also in the process right now of forming a local organizing committee. So for those of you in the local DC area, I'll be happy to hear if you are interested in being part of this effort. And with that, I'll end my presentation and certainly thank members of my team, Pasley and Dimitri and Chris, and certainly a number of program officers from a number of institutes and centers at the NIH, as well as our agency partners, the FDA, the EPA, NASA, BARDA, the VA, and also the International Space Station and the Center for Advancement of Science and Space, as well as a number of pharmaceutical companies that makes up the IQ MPS affiliate. So thank you very much, and we'll stay around for during the panel discussion. So turning it back to Elena.
Thank you, Dr. Tagle. This was amazing. I mean, it's such amazing to see what have happened over such a few years, you know, that all this progress has been made. I'm just blown away. And it's opening up new opportunities. Of course, we can do so many new things. And one of the questions I will encourage you all to write your questions in the Q&A box, but talking about validation and qualification, which is, of course, really, really important to be able to use these methods and this new approach methodologies. And with that leads me to the second presenter, which is Dr. Ivan Rusin, he's a professor at the Department of Veterinary Integrative Biosciences and the principal investigator in the Laboratory of Environmental Genomics at the Texas A&M University. His lab has an active research portfolio funded by the NIH and the US EPA with a focus on the mechanism of action of environmental toxicants and the genetic factors of the susceptibility to toxicant-induced injury. Though Dr. Rusin has conducted excellent basic research activities, his talk today will focus more on something else that is very important specifically for this project, which is validation and qualification of MPS, where he's leading the TexVA, and that's what we will hear about now. So go ahead, Dr. Rusin.
Thank you, Helena, for an invitation, and thank you, our colleagues at ICCVAM, for organizing this event, and thank you all for joining. One correction to my introduction: we're no longer funded by EPA. 2 grants that we had were terminated about a month ago. So what I wanted to talk about is to pick up where Dr. Tagle has kind of left off, which is the landscape of development of microphysiological systems in the United States. And he described the US landscape, and equally impressive developments are happening also in many Asian countries, in European Union and other parts of the world. But while he kind of showed more on the development, I wanted to focus more on this part, which is the building confidence in the microphysiological systems, and again acknowledge NCAT's foresight to not only invest into development of a variety of these models, but also in trying to think about how these can be used by the end users and what type of confidence level they can have. And as you have heard from him, the development of these models all the way from back in around 2010, 2012, when NIH and FDA and DARPA joined together to fund the first implementation of this. This is not slowing down, and with the complementary program in US and upcoming Horizon programs in Europe, and also investments in Korea, Japan, and China, the development of these models will continue. And it's really, staggering to know that it's, hundreds of millions of dollars have been invested on in the US, and equal amounts, if not larger, in the rest of the world as well. So again, there's a lot of this technology out there. And as you've seen from the videos that Dr. Tagle played, this is is really cool stuff. And the gadgets that are produced are very unique, very exciting. And not surprisingly, a lot of this makes it into the press. And on the left, what you can see is a news item from 2015, where, again, this excitement already has reached the news wires, where the promise was for this technology to be cheaper, faster, and more accurate and more humane in use for drug and chemical safety evaluations. And this is more recent, from around 2020. Dr. Ingber, one of the pioneers in this field and the founder of Emulate, he's not mincing words when he's saying that their job is to make science fiction into science fact. So these are very exciting developments, and I think both researchers and regulators and end users and general public are really interested in understanding how this technology can actually makes its way into the reality. Now, while I'll touch on the more recent announcements, back about 3 or 4 years ago, when the Food and Drug Omnibus Reform Act, or FDORA of 2020, was passed, a variety of news wires, and this is one example from NPR, announced that FDA no longer requires all drugs to be tested on animals before human trials. So I think what's important to recognize is that, some of these, facts are taken out of context sometimes, especially when the media is involved. And as Dr. Bibinek, who's on the call today, pointed out in a couple of presentations, the statutory language never required animal studies for drug development. In the previous text, it was mentioned that these are preclinical tests, including tests on animals. And in the FDORA 2020, this is just a longer explanation of non-clinical tests, and this includes cell-based assays, MPS, computer modeling, other test methods, and animal tests are still an option. And in addition to the news announcements that were shown by Dr. Tagley, again, I wanted to point out the FDA news release. The commissioner, said that the FDA is interested in leveraging AI-based computer modeling, MPS, and other advances in drug development. And again, some of the news that came out stated that FDA announces plan to phase out animal testing requirement, which is not a requirement, but also some have missed the second part of this title, which is for monoclonal antibodies and other drugs that are, protein or other complex therapeutics, not small molecules. And Dr. Villeneuve will, show us how the pharmaceutical company has actually been approaching this, for quite some time now. So it's great that the agencies are making these announcements, but this is not the only impetus for this technology to be made into the reality. So as Dr. Tagley mentioned, this ecosystem of businesses that offer this technology as a, for sale or as a service has really exploded in the last 10 years. And this is just a small, list of companies that are offering various technologies. And it's fantastic that the offerings are so large, and there is an equal, if not larger, number of academic labs that actually develop various technologies that could be used in real world for drug and chemical safety testing. But from the endpoint or standpoint of the user of this technology, whether it's a toxicologist who's working at the drug company or a food company or a chemical company, or a regulator who is interested in considering these data as part of the safety or efficacy package, the challenges are daunting because there's almost too much availability and also very little in terms of understanding how robust and reproducible these models are. So on the one hand, definitely in the last 15 years with NCATS and other investments, both in the US and abroad, we have built it. Now, the question is whether the users will come. And, the questions, are twofold, in my opinion. One is, whether it's a technology that can be used or understood, whether it's sometimes too complicated for the end user. So akin the baby and the computer. Obviously, babies learn how to use computers very fast, but there is a learning curve. And the second point is, how does one actually understand the utility, the cost, the reliability, reproducibility of these models without actually relying necessarily only on the vendors themselves providing this information. So as already mentioned in Dr. Tagley's presentation, we have had funding for two-year cycles for the Tissue Chip Testing Center from 2016 to 2020. And our remit at that time was to take MPS that were developed with funding from NCATS and bring them to a independent laboratory and to try to replicate some of the work that was published by the developers, but also to see how these technologies can be used in kind of toxicology applications. So because, as I mentioned, these were 2-year cycles, and we had to work with a very large number of collaborators, and my again heart goes out not only to our collaborators who are listed here, but also to all the folks in my lab and our collaborators in Houston who work with us on this. It's really, quite a non-academic exercise to try to take maybe on average 6 months per tissue chip technology, and first to establish these collaborative research agreements, to test these devices, to reproduce what was done by the developers, and then to try to see whether we can start defining context of use and conducting additional studies, and also making these data publicly available for others. So what kind of we learned from this? The first question one may ask is, did we get these academic lab made devices to work outside of the developer lab? And I can say that mostly yes. It's remarkable. I mean, the success was not 100%, but it's great that many of these are robust and reproducible. We were able to replicate the results from the developers. Again, mostly yes. Again, the success it means different things in this space to different people. The biggest challenge to technology transfer was really the cells rather than devices themselves. And some of the time when we could not reproduce exactly the findings that were reported was because we had cells from a different donor or a different cell line. Now, were these devices ready for testing drug safety in the real world? And I can, again, confidently say that most were not. And again, there is time to move this technology from academic lab to the actual market, to then utility of use by the end users, and then to regulatory decisions. And the most important learning from these studies was that really the developers learned probably much more than we did, because they were able to recognize what the pitfalls these devices have and how to improve them. So in all of our opinions, this was a successful exercise, but the requirement from NCATS in the second round of funding was that we had to present a business plan for revenue generating activity, meaning that we had to take this rapid robustness and reproducibility testing outside of being dependent on NIH funding. So what we've done, we've kind of went around and talked to a number of our colleagues across the board, and government agencies and companies of different type, and try to recruit them into a consortium. And the goal was to bring together not just pharma companies, but also a very diverse set of individuals and companies with different interests, and then ask those consortium members to develop a work plan to identify the tissue chips, the organs, the platforms, the cells, the chemicals, the phenotypes. They're the end users. They need to be telling us what we need to test. And then our job was to execute on this work plan and to procure equipment to establish these models, to produce the data, to share these data, and then to, again, inform our consortium members in the larger world about the technology readiness to be used for real-world applications. So the value proposition is pretty straightforward. As Dr. Tagley mentioned, this is a pay-to-play model. The funding is provided by consortium members. For the last 5 years, it has been at exactly the same level, $100,000 per year per member. Texas A&M University considers this scientific service, so it charges 0% overhead, and faculty salaries are not included in the expenses that we can charge to the consortium. We ask our members to contribute several scientists who will participate in the meetings that we have, and to have this input on the annual work plan. What we provide is data protocols. We make all the data and protocols publicly available. After 1 year of the consortium's calendar year, we engage quite a bit, not only individually with consortium members, but they talk to each other. We provide unlimited technical and scientific support. We've seen more than 50 different MPS models in the last 8 years. We've been doing this work, and then we publish. So one of the needs from the end users is that, they're excited about this technology, but they really don't have the capacity to test every possible vendor's chips in their own labs. And so this trust but verify can be done as a consortium. So this is one example of, published data from company Mimitas. They developed a liver on the chip technology in collaboration with the University of Pittsburgh. And this is done in a 96 kind of device format. So this is organoplate 2-lane 96 version. And they published quite a bit of data. In red is the data from their publication, and the analysis of putting this into the the classical 2x2 classification metrics was done by us and also published in 2022. So what you can see on the top is that they tested 162 chemicals in this device, but to enable that throughput, they only did one concentration, one time point for each of the compounds. And they selected these compounds based on other publications, so they actually included data from these other publications on DILI, drug-induced liver injury prediction. And when you actually compare their data to those other publications, it's actually not, a better model, so to speak, to predict DILI. They also had another experiment where they did concentration response on a smaller number of compounds. And again, there were comparator data provided in their own publication from other models, much simpler two-dimensional primary human hepatocytes. iPS-derived cells have G2s and whatnot. And then what you can see is when you do dose response and actually your throughput drops, you can get close to the same kind of prediction metrics than the other technologies are. Well, the big difference is that there's probably a multiple of 10 on the cost, going from a simple system to this particular system. And I understand they're no longer marketing this platform, but that's just, trust but verify. Someone publishes a paper and says we have a model, but then benchmarking this model to other models is actually not straightforward. And the communications medicine paper that Dr. Tagle showed from Emulate, used Emulate chip and used a smaller number of compounds. And again, they published their data on prediction of drug-induced liver injury. And again, it's a remarkable achievement. But when we took their data and put it on the same kind of scale with the other publications, you see that Emulate model is among one of the best that is available. But again, there is quite a significant cost and throughput difference from the simple technologies. So again, doing this at every company, every end user, every regulatory agency is virtually impossible. So our consortium offers the ability to do this all together. So we started in 2020. COVID was an interesting year, but we still done a lot of work. We still had some funding from NCATS, and we recruited 2 pharma companies, US EPA, American Chemistry Council, and Division of the National Toxicology Program from NIEHS, NIH. And then, the membership is 1 calendar year. There are no long-term contracts. We have added, a consumer goods and food company, Unilever, and a German Merck, which is, a pharmaceutical, but also a chemical company in 2021. In 2023, Bristol Myers Squibb had some financial reorganization, and they had to cut all of the academic memberships, but we added Roche. In 2024, EPA also had quite a significant cut to their research budget, and then American Chemistry Council, this year also had to withdraw for financial reasons. So we were able to stay at about 7 members throughout. And again, folks leave for financial reasons primarily, but we're also adding new members, and we're recruiting new members this year as well for next year. So every year, as I mentioned, our members are talking to us about what they would like to test. And this is kind of a summary of these planning meetings that we have at the beginning of the year. So you can recall in 2020, we had 5 members, and since 2021, we had 7 members. And these are the organs or tissues that are of great interest to individual members. And the numbers represent the number of consortium members who have expressed interest in that particular organ or tissue. So you can see that liver and kidney has been, a topic of great interest throughout, blood-brain barrier and some of the years, GI in some of the years, lung in some of the years. Some other models or organs are mentioned by one or two members, but then ultimately, as we decide what is actually realistic for us to accomplish in 1 year, only things that are highlighted in green are actually being tested. I highlighted in yellow our interest in MPS models of reproductive and developmental outcome, and these are funded by other mechanisms, but we're sharing all this data with our consortium members as well before we publish on them. So back to value proposition. So again, we collect anywhere from $500,000 to $700,000 a year in membership fees, and how much work we actually are doing. In 2020, we ran 1,500 microphysiological systems or chips. So these are the number of conditions that were tested, and about equal number of comparator 2D models has been tested as well. So as you can see, depending on the complexity and the types of organs and the platforms that we're using, the numbers fluctuate. And then last year was really a very large year, but again, not all of the models are truly these very complex devices run one at a time. Some of the platforms are 96-well, trans-well, that also we consider complex in vitro model. So again, we're producing quite a bit of data for a price that each individual consortium member cannot really afford to do anything at that scale. And our annual work plans are consensus based, meaning that every one of our consortium members has something to gain or has an interest in our work plan. So what we view this as evidence-based qualification by TaxVal Consortium. So we compare industry standard, which is frequently a 2D model, to the new MPS, and frequently we compare different MPS to each other, which has cells from different vendors and donors. We're trying to phenotype in a very sensible and economically responsible way. So we're doing a lot of imaging, we're doing a lot of biochemical, analytical chemistry. We're doing some limited, omics data, primarily high throughput genomics. So many people ask, can you do metabolomics? Can you do proteomics? The answer is yes, but it's a matter of whether we want to kind of go wide or tall in some of these designs, and then our consortium members really guide us as to what is the most sensible and probably most realistic way of phenotyping these models and how they will actually use them in their own labs. Now, the cost-benefit analysis for setup and operation is really different from just the cost of the acquisition of these models. There are upfront costs, but also there are operation costs and there are costs to phenotype, and all of this is being tabulated and given to our consortium members. And then we're trying to make sure that we're not only testing drugs, we're also testing, industrial chemicals and chemicals of different types. So we're trying to keep domain of applicability broad. So this is one example of, again, this, value proposition or cost-benefit analysis. This is an example of a blood-brain barrier model. So we have, done brain microvascular endothelial cells ourselves from published protocols. We can put them into 24 or 96 well trans wells. We can put them into a CN Bio, 12 device platform with perfusion. We can buy these, cells from a commercial vendor, and we can combine brain microvascular endothelial cells with astrocytes and parasites. So again, there's a lot of dimension in this, experimental design, and I'll spare you the details, but basically I'll conclude that all of these models produce about the same level of barrier, and all of these models produce about the same type of utility or how long that barrier is actually existing. I would say that Fujifilm CDI blood-brain barrier kit, achieves that, about 1 week earlier than our other technologies. So that may be some cost saving. But in the last column, you can see the cost per sample. So again, the barrier is about the same quality, but it can cost you anywhere from $7 to $450 to actually achieve the same biology, but in a slightly different design. And what we also found is that, adding perfusion in this particular model does not really afford any additional benefits. So again, this cost goes well beyond, cost analysis goes well beyond just robustness and reproducibility. So I'll give you a couple of examples of what we've been working on in the last 2 years, just to give you again an example of diversity of work that we do. In 2024, we had a lot of work to complete, an experiment that was a spillover from 2023. We decided to test 4 different organs, and then within each organ to test the same compounds, and then to compare 2D to different microphysiological systems to each other. So again, just to spare you the details, the analytical chemistry alone was 10,000 samples for the analysis. We are very interested in kind of simpler models of renal transport. There are lots of very unique liver models, especially vascularized liver models. So we tested different models like that. And again, the numbers, the pricing on the right is about 1 year old. So these costs have increased since then. But you can see the design is about the same, but the cost per chip is different from $6 to $25. And again, the number of cells is also different. The ease of operation is very different. So there's quite a bit of significant difference in the pricing when maybe the physiology that you're modeling is very similar. We're interested in cholestatic liver injury model. We're interested in cross-species collaborations. So this is really not charged to TaxVal because one of our members, Unilever, has a no animal cells policies, but we still do these experiments and share them with appropriate members of the consortium. And then we're also taking part in this, pharma ring trial of one of the liver models from tissues as well. This year, the work is, quite diverse. We're working on liver, vascularized models, kidney, lung, also 2 organ systems. We're using quite a number of different models, and then we're asking very, very different questions. So again, we really are, happy that our consortium members are helping us through that. So, to conclude, is this consortium a success? In my opinion, it is, and obviously because our consortium members are continuing in their participation, it is also the answer to them as well. It's a very robust collaboration of very diverse stakeholders, and to them the value proposition does exist for try before you buy operations through TaxVal. So there are a number of learnings of the consortium, rather than the actual data. So for example, we all learn together how to best select a model for testing. Again, you've seen how much diversity is there in offerings. So now we're all learning from each other and our data as to which models are best for which context. We're also learning what MPS, how MPS can be used. Some can be used for maybe disease modeling, others for toxicology testing. Some can and others cannot be used for ADME PK contexts. Some can be used for barrier function, others for different functionalities. And then also a very big consideration is reproducible cells for each of these MPS. But also there is, the cost component to this, the phenotyping method comparison to see what is actually the best performance, understanding how easy and expensive it is to continuously operate these models in the lab, and also what other equipment is needed. So, I frequently use this all that glitters is not gold. And this is very, very exciting science. But what we're trying to do the hard labor of some sort of testing and qualification of these before they make their way into the real world. And to us, success does not necessarily mean that this is, we prove that the model works. To us, success means that our consortium members decide to onboard or decide not to onboard this MPS in their lab. And if they decide to do that, we'll help them. I'll leave you with publications that we had. This is, a sampling of publications that we've done on this work of testing and reproducibility and robustness. And the publication that is highlighted is more of a perspective from the consortium. And this slide will be available for everyone. So with that, I'll conclude and participate in the question and answer later. Thank you.
Thank you, Dr. Rosen. Also very impressive work and very, very important to understand when the context of use of these ones, which is actually where we're going to go into now as well. The last speaker is Dr. Remy Vellanu. He's lab head at the Roche, previously working with Emulate. He has a background in airway physiology, and his research prior to joining Roche was also focused on modeling airway tissue using 3D cell culture systems, organs on chips, and disease models, including asthma, COPD, and viral infections. Now, when he is at Roche, he's also leveraging these advanced in vitro systems to improve drug development processes. And we will hear a little bit on actual application of these models, pros and cons, etc. So with that, please go ahead. And again, Q&A, we're not going to, I saw someone was trying to raise their hands. We're not going to unmute you, but please put your question in the Q&A and we will come to them after this last presentation. Please go ahead. Dr. Villeneuve.
Thank you very much. Do you see my slide in full screen?
Yes, we do.
Is it okay? Okay, super.
Looks good.
Okay, so I'm not used to Teams too much, so please forgive me if there's any little issue. But essentially, what I'm going to show to you today is a bit our experience with these tools that have been presented by both presenters before, and show you how we envision the future of using these tools, and maybe talk about their their potential and also limitation that we see. So that's our end user perspective on the topic. And maybe to have everyone on the same page, these tools, these prediction tools have been given a lot of different names. And in this presentation, what I'm going to do is show you, show you what definition we'll use across this presentation. So NAMs, or new approach methodologies, essentially an FDA defined term, which in this case includes novel in vitro, in silico, as well as selected in vivo methods that are used to assess the safety of drugs. And one important point here I want to make is that according to this definition, and I think that's the latest definition, it hasn't changed, I think, is in vivo methods are also considered NAMs when they improve the predictivity, shift studies to lower animal, or help replace, reduce, and refine animal use in development programs. So it's an important point. And this, in this case, we're talking about mostly humanized animals or genetically engineering mouse. So in the recent years, as it's been pointed out in the previous presentation, regulators have made several announcements regarding the use of NAMs for safety assessment. And this was really reflecting a shift in mindset and growing openness to evaluate this, the readiness of these decision tools to support the risk-benefit assessment across the industry. And this obviously culminated recently with the announcement of a new FDA roadmap, as it's been pointed out, on reducing animal testing for preclinical safety studies, with several caveats as well mentioned before. And that's really signaled a real momentum for the exploration of NAMs in the pharma industry. But I really want to point out here that NAMs, or complex in vitro models, were already used in the industry for quite some time already. And I would say maybe a bit more than a decade now, with really dozens of platforms that we have tested over time, from very simple models to very complex models, really looking for compelling use cases. So the academic research produced a lot of these tools, and what we did is really try to evaluate each individual tools according to our different context of use, and see what we could get out of them. And currently, the situation we're in is that a lot of our programs are supported by these NAMs. So majority of the programs still use conventional animal models, but a growing proportion of these programs use what we call NAMs augmented approach, which is a combination of conventional methods and the use of these more complex in vitro systems. As well as what I want to point out and talk a bit about in the coming slides is we also have this NAMs only approach, where at least for safety assessment, no conventional animal methods is used. And this is mostly in the absence of relevant animal species. So this is really driven by a scientific question. And what we think is in the future that we think that NAMs will be increasingly used for portfolio support. At least that's the sense the industry is going towards, due to several reasons. And I listed a few here. For example, modalities of drugs that are becoming more and more complex. So we have a lot of experience with small molecules, a bit less with more new modalities such as large molecules, antibodies, gene therapy, for instance, or nucleic acids, and so on and so forth, where the specific mode of action of this molecule is very human specific. So much more difficult to test in animals, and where the cross-reactivity is questionable due to maybe the expression of the target is different, or the functionality is different, or the tissue distribution of the target is different in preclinical species, and as well as the incentive from the regulators that pushes us as well towards the use of these tools. And what I want to show you here is more precisely our experience. So this is really programs that we had in the past, let's say in the past 5 years. And this is for a safety assessment. So when we assess the safety of our molecules prior entry into human, and how we develop the package for entry into human. And as we can see here, we have around 10 programs, now a bit more, and where we have this in vitro only approach that I just described. So for the safety assessment of these molecules, we only used in vitro models. No animal was used there. We also, if going into a little bit more detail, we use some of the readout from efficacy models for the safety as well and for the PK. But for the safety, mostly the assessment was done solely using in vitro systems. And you have here a different justification. What's important to note here, as I say, is that these were done already some time ago, so before the different announcement from the regulators when scientifically justified. The NAMs focused approach for entry into human, so far in our experience, were only for biologics, so antibodies, where you have generally a high specificity for the target and the low off-target risks. And as I said, the NAMs approach were not fully animal-free. Animals were still used for pharmacology and PK, so that was really restricted to safety assessment. And there were no requests received to use cross-reactive homologous surrogates, so which are molecules that are specifically synthesized to be cross-reactive with animal. This was not requested by the health authorities, so everything was accepted as is by the health authorities, which is an important point I wanted to make. What's interesting to note as well is the narrow scope of these studies. They are all in the absence of relevant species in the oncology field. So that's really where the health authorities accept these studies is when the indication is really life-threatening. So this is where maybe the bar is a bit lower for the acceptance of these tools. To my knowledge, there haven't been too many accepted NAMs only approaches outside of oncologies, but I think this is coming, and I will show you some data about that as well later. One important thing to note as well, and this is really something we've been facing in the past, the starting dose is often quite low. So we start with a MABEL approach, and this is due to the lack of confidence in this tool yet to predict exactly a human dose. So we start low, and then we ramp up the dose. And the translatability of these NAMs for clinical outcomes needs still to be established. It takes time, but we still need to publish, generate use cases, publish these use cases, share with the community, and also inform the regulators on the predictivity of these methods. So that's why it takes so long to apply these tools. What I want to show you here is, so I show you our experience before, but I want to also expand a bit to a recent survey that has been published in the recent months we've contributed to. And I think it's interesting here because it shows beyond Roche, so beyond our experience, what is the perspective on other pharma as well. So here we run a survey. This was done through BioSafe, NAMS task force. Essentially, it's a modality agnostic 12 question survey, which was also biologic focused with 78 questions around case studies that we sent through 288 unique entities. So 188 companies, and I think 27 responded to the survey. 9 of those companies had NAMs accepted by regulators to replace animal testing, and had experience with this path across multiple regions. So not only FDA or EMA, but also across different regulators. In addition, most respondents indicated that they would consider using a NAM-based approach in the future to replace large animal testing. So, which is quite encouraging for the field. So we're not the only one. A lot of different pharma are going that route. You have this graph as well on the side, the justification to use NAMs, and you can see a bit similar to what I just told you to our experience, and it's not surprising. Every pharma have a similar experience. The lack of pharmacologically relevant species is a justification to use NAMs. Improved translatability, essentially having a human-based system is of value here. Scientific justification, 3 R's, so there is also a justification to lower the use of animals or complement the use of animals. You can see as well a shortened timeline, although to me this is still unclear if using NAMs will shorten any timeline. That is, at least now we are still in the situation where we explore and we are okay spending a bit more time and resources to use NAMs in our programs. And also add mechanistic insights, so context for finding in animal clinic. This is really important for mechanistic safety, where we have a finding in animal or in the clinic, and we use NAMs to explore this, recapitulate, model this adverse events. So this is how NAMs are used across pharma at the moment, I would say. I want to link it to this other paper as well. You can flash the QR code here to access this paper, which was also quite a nice publication highlighting use cases of NAMs across the industry. And what is nice here, we have these categories that were defined to think about this topic, which is quite complex. But we have defined a few categories to be able to evaluate different use cases of NAMs. And what's maybe I will merge both of the publication on these slides, where you can see here first the framework, the NAM safety assessment framework that we came up with. And what you can see, this is interesting to have at a glance a way to think about the topic. So you can see that the gradient between the green category and the red category highlight the acceptance by health authorities. So as I show you, the health authorities are keen to accept NAMs to replace animal testing in the context of biologics when the target specificity is high. In oncology, when there is no cross-reactivity or no pharmacological relevant animal species. So that's really the use case that is now pretty much accepted by health authorities. When we move towards the the right of the gradient, the table, we have biologics which have relevant pharmacologically relevant animal species, but the mode of action of the molecule is clinically validated. What does that mean? Is that we have a lot of experience with the target. It can be a checkpoint inhibitor, it can be a CD20 antibody, where we know a lot. We have a good way of evidence about the safety of the molecule, and there that's where the health authorities now become more open to accept NAMs in the absence of animal testing. And then there is a biologic still, but there is a presence of animal species, and it's a new target or mode of action. This is a case where I don't think health authorities still yet accept NAMs for in replacement of animal models. And of course, small molecules and maybe new modalities. This is a very high bar because animal models have been used for decades to assess to predict a human response, and this is where NAMs are still not accepted. And I think it will take some time to use NAM to replace animal in this context here. Non-surprisingly, this is the result of the BioSafe survey I just showed you. Most of the use cases here, you can see the use case at the bottom, the numbers of use cases are in this, let's say, green / yellow zone of the framework, which is mostly cancer essentially, so antibodies, advanced cancers, cell therapies as well. You have a few use cases now outside of oncology that are rare. So you have a virology, respiratory disease, hemophilia, but this is really not the norm at the moment. But maybe some pioneering cases that can open the door for NAM use to replace animal species and animal testing. There are still, although there are still a barrier to NAM use, although the industry is adopting these tools more and more, and health authorities are accepting them. There are perceived barriers to NAM use, and I think it's important to note that respondents, at least from this survey I just showed you, and this mirror as well our internal experience, that regulatory acceptance by FDA is a big barrier. Although as you saw, some are accepted, but really that's a minority. Lack of harmonized global regulatory acceptance. It's true that different health authorities have different perspective on the topic, and there is still lacking this harmonization, this global harmonization on NAM's acceptance. Concern about translatability, added time for the qualification and validation of novel NAMs. This means resources for the industry to spend, although consortia such as the one Dr. Rustin just showed can help there to validate and qualify some NAMs for specific context of use. And concern about false positive negative, which is also another way to mention this translatability and predictivity. And all this is around the validation topic, right? So in blue, I highlighted what I think fall under this scope of validation, and this is a big effort that is ongoing at the moment, and I think, in my humble opinion, where resources should be put in the future. So in conclusion, what I wanted to say is really that when scientifically justified, NAMs-based submissions are accepted globally by regulators. Most of the case in a narrow scope of oncology, a large molecule, in the absence of relevant animal species, but still it's accepted. Opportunity exists to scientifically justify NAMs-based approach in a broader setting. This is what we are doing at the moment at Roche as well, is to expand beyond this narrow use case of oncology and go beyond to more indications and use cases. However, there are really remaining concerns about global regulatory harmonization and clinical translatability. And the validation really is a key here, and very happy to contribute to this workshop, because what ICCVAM is doing as well there is really important. This really need for validation of alternative methods is the only way, the only path to reduce animal use in the future. And so I think I'm going to stop there. There's a lot to say, but maybe we can talk about other topics in the Q&A session, and happy to stay later for answering any question. Thank you very much.
Thank you, Villano. I'm just gonna take the time to. It was again excellent, and really nice to see also where we need to put more efforts. And I have to say, I'm very happy that ICCVAM and ICCVAM are working on several of those points that you were pointing out there. So I want to now introduce Dr. Emily Renke, that is the principal investigator at NICEDAM's support contract, and Dr. Kamal Mansouri, which is NICEDAM's acting deputy director, to moderate the Q&A. So there are some Q&A there, so I'll hand it over to you guys. And, thanks you again, all the speakers.
Thanks a lot, Helena, and thank you to the speakers for the great presentations, and thank you to all the attendees who stayed with us till the end of this communities of practice. So I'll start with this question that might be for Dr. Villeneuve, but can be answered by any one of the speakers. The question says, has the tissue chip model been applied for immuno-oncology drug development, such as monoclonal AB blocking checkpoints or B-specific ABS?
Sure, I can maybe take this question first, and then leave my colleague maybe answer further. Actually, mostly for immuno-oncology, in our experience. That's where most of the cases I just showed were immuno-oncology, for bispecifics as well, and checkpoint inhibitor. Yeah, so I think this is where we have a lot of expertise, a lot of knowledge about at the moment, where animal species are really limited because the target is simply not present most of the case. So it's a very good use cases for exploring and adopting NAMs. So yeah, that's a good use case for NAMs.
Well, if I can add, this is a great example of once you have confidence in the platform itself, you can then start, thinking of how to use it. The challenge that we had in our consortium and trying to bring in new modalities into qualification or testing that we're doing is the lack of a robust set of molecules with known positive and negative effects. So while it's fairly easy to find small molecules, both drugs and non-pharmaceuticals for testing robustness and reproducibility, in the last couple of years, when the topic of new drug modalities or different molecules has come up, we realized that we really don't have a good set of non-proprietary molecules to put in. So this is one challenge going forward, but still each individual consortium member and the user can then, once they believe in the technology, they can actually do this work in-house. But the ability to qualify is limited to using these modalities.
Thank you both. I think that I'm actually going to go ahead and do a combination of a couple of questions, because they're all kind of hitting on the same point. I'm going to throw a monkey wrench on my moderator. Sorry, Kamal. This was a nice segue into the concept around, we've had a couple of questions around validation. What do you do when you have multiple academic facilities who are all developing different approaches to maybe the same organ system? So what are some of the ways that any of you would foresee as a way to limit the need for comprehensive validation across every single platform, or is that going to be necessary across every single organ subtype? Or and then also, what do you do when you have, how are you envisioning validation going forward, given the limitation of the number of small molecules that we have? Sorry, I'm just throwing a hard one out there at y'all.
Well, let me start. This is a very important concern. So in the NCATS funded for years, our remit was to take focus on platforms that were developed with NCATS funding. And we were able to recruit about half of the grantees who were reached out to into the testing routine. On the consortium, we do not dictate the platforms that the consortium members would like to put onto the working plan every year, but inevitably 95, I think, % of all the suggestions have been commercial platforms, and especially platforms that are quite robust in terms of their offerings, platforms that do not require CRO-like testing, meaning that these can be given to the end user, and the end user can decide what to do with them, because a number of the MPS companies are realizing that they want to really be in the contract research organization business. So it's impossible to test every platform, but the way our consortium has been thinking about popularizing and sharing the information is the study design, sharing all of our experiences and both advantages and limitations, and they're not usually MPS dependent. They can be generalizable. So we, while not doing the round robin validation right now, and that's impossible, we're focusing on the big questions, on defining what should be measured, defining what chemicals should be used, and defining what the success looks like, and when do you decide to improve on something. So there's learnings, but there's not really an OECD like round robin validation that our consortium is doing, and that's not realistic.
So kind of thinking more around the lines of like a performance standard around different organ types. All right, thank you, Kamel. I'll pass it back to you.
Thank you, Emily. So again, I just want to remind everyone of the attendees, if you have a question, to please post it on the Q&A section. I will go probably to one of the upvoted questions. How realistic do you think the proposed timeline for the FDA roadmap for reducing animal testing is? 3 to 5 years, it says.
Maybe I can give a bit of perspective on that from end user. I mean, the reality is that we are already replacing some animals using these tools. Likely, it's the use case are going to grow, especially with the help of this momentum that we've seen recently. I think giving a timeline is impossible. I cannot give you a timeline. 3 to 5 years seems optimistic to me. Now, if you ask me personally, when we're gonna fully replace animals with these systems, I would say more 30 years or 50 years. That would be my honest answer. But I'm maybe pessimistic here. I'm a pessimistic person, or maybe a realistic person. So maybe it's going to be much before that. But it's really difficult to give a precise timeline. What I can tell you is really both the industry, the pharma industry, the manufacturers, and academia are working hard towards testing these systems, and we have the safety of our patient in mind first and foremost. So we will not jeopardize that with suboptimal methods. We'll use what method is the best to ensure the safety of the patient. So that's really our driving principle. If it's a non-perfect, if it's an animal, we'll go with it as well.
So thank you, Remy. Just to add perspective from international point of view. So I was a couple of weeks ago in Helsinki at the European Commission's workshop on the roadmap of replacing animal testing and chemical safety assessments. So in Europe, this topic is equally acute, if not more acute. And as many of the participants may know, that there is a ban of using animals for testing of cosmetic ingredients, and that ban has been around for quite some time, and that was an example of setting a hard and fast deadline and then seeing what happens. And without going into the discussion what happened, I can tell you that in that roadmap workshop, the Commission has announced that they will release the roadmap sometime later this year or early in 2026, and that roadmap will not contain any deadlines, even though the amount of pressure that the European regulators are under in terms of this topic is much greater than here on this side of the Atlantic. And during that workshop, a number of people have asked the Commission to commit to a deadline, and the Commission has very forcefully declined that, saying that this is not a useful way of defining a flip switch type moment. So as Remy said, this is not a new topic. This is a topic that everyone has been hard at work on, and this is going to be a gradual transition. And I think what's important for all of us to keep in mind is the safety of both consumers and people and patients, and no one wants to advance something that is unsafe. And the current practices are ensuring safety. Yes, there are examples of bad failures, but those bad failures are few and far between compared to the number of chemicals and drugs on the market that have been safely put on the market. So again, I would encourage everyone not to think about the switch, but think about how we can all combine our forces to find case examples and to promote the use of alternatives when it is safe to do so.
Yeah, and just wanted to add from the NIH perspective, it's certainly more, kind of like what I mentioned in my presentation on complementary. It's really finding an area or a niche for NAMs where animal models either do not exist or have consistently performed poorly, and that I think is where NAMs can be positioned and make much more headway in biomedical research and in safety assessments. Yeah, but having said that, I think there are going to be certainly some specific contexts of use where NAMs would replace animals, but not completely eliminate animal testing.
Okay. Thank you for that. This leads actually very nicely into our next question, kind of around, shortfalls and where NAMs can maybe come in or maybe where they can't. There's skepticism around using these in PK/PD studies because of multi-organ involvement. Can each organ chip be connected to make up a multi-organ system? How does culture media contribute to result interpretation around that? So, kind of looking for what the approaches are to doing the multi-organ approaches.
Maybe I can start, perhaps I don't know if anybody has a better experience with that. I was part of a few programs on organ linking in the past, and I think what's important to point here is that these technologies are relatively complex. I mean, it's not just taking some tubings, connected 5 chips, and then you have a mini human on a chip. It's really complex. I think it should not be underestimated. And the in vitro artifacts are numerous. It's quite a challenging topic technically and scientifically. So I think it's a promising direction, but at the moment I would say we are not using this internally. This I can say. We haven't been necessarily very convinced by some linked in vitro systems. Maybe this is much more common in in silico modeling, where you have some great PK model, but in vitro is relatively challenging. Yeah, that's my perspective on that.
Yeah, I certainly would agree with Remy that in terms of in silico, certainly PBPK modeling would be something that can be used. But at the same time, there are in vitro models, even commercially available ones that uses and commercialize multi-organ platforms such as the ones being marketed by Tissues and Hesperos?
Yeah, so a couple of points here. One is MPS are really, not a particular challenge that we have in translating from in vitro platform, any type of in vitro platform to in vivo. Going from, a concentration in a dish to oral equivalent dose or inhaled dose or, skin absorption is not straightforward. And also the use of in vitro data to parameterize, PK or PBK models, has decades of experience with, I think, more successes than failures. So adding an MPS where you truly are trying to, say that you have a liver and a gut and a kidney together is a big if. It's not like we have not been able to use in vitro data and models to actually very accurately predict PK properties of, most chemicals. Obviously, there are a number of exceptions to that. So the question is not, can it be done with MPS? It can be done, and NCATS has funded, a program, and there were a number of very elegant publications to that effect. The question really for the end users and regulators is it practical to use these models every day or at least once a month? And that's a big if. And so that's where there is a gap right now from theoretically, you can have multi-organ systems, CN Bio, Tissues, Hesperos, and others, but the level of confidence in those models is currently low. But there are big efforts, both internationally and also domestically, to address this gap. So U.S. chemical and food regulators and those in Europe and in Japan and Korea are talking about designing a joint study. It probably cannot be called a ring trial on a 2-organ system. But what we faced most challenge with in that particular design is not finding the models that can be used. There are plenty of MPS that can do 2, 3, 4 things together. The biggest challenge is finding the right chemicals. Are we testing drugs? Are we testing environmental compounds? And do we have the right human data to benchmark them? That's number one. Number two is the throughput of these models. So those are very practical limitations, and we are possibly 3 to 5 years away from having confidence in 2-organ systems. Not just are they available, they are available. Do we have confidence? That's probably will take 3 to 5 years alone.
Thanks a lot. So the next question is also a little bit about opinion and how we can tackle education in general. So the question says, will more effort be put into educating the public on context of use and limitations of NAMs where animal models are not available? Seems like animal studies are being demonized as being outdated and of little use or even misleading, and NAMs presented as ready to go should be all we use. This deception will likely make the public less trusting of scientific organizations when it takes much more longer to get NAMs to where they need to be.
I can maybe start on that. I'm sure my colleagues have a lot to say as well, but I think that's an important point, and I think it's a conundrum a bit where, yes, if you oversell NAMs as a solution for replacing animals, you may deceive the community and therefore maybe hamper their adoption. However, I think each players have their role to play. I was in organization where we kind of were selling these systems, right? We were maybe even overselling the system, but thanks to that, a lot of resource has been put in testing these systems, and maybe it brought us to this point where regulators are looking into it more carefully, and so I think if each players, I'm talking academia, regulatory, manufacturers, end user industry, play their role in a fair way, I think we're we're going to end up in a good place. I think overselling a bit is fine. I was against it at the beginning. I thought that it would set the wrong expectations, but at the same time, I noticed how powerful it was in securing resources internally, securing this momentum we're seeing. So it's a fine line, but I think it's fine to market a bit these tools. Of course, not going too far, but it's needed to create this momentum, and that's what I realized. Although I was strongly against it before, I think it has a place there.
Yeah, I agree with Remy in the sense of managing expectations well. I think clearly we need to set aspirational goals to set a trajectory of where we want the technology to be and its use. But at the same time, we need to be rooted in terms of what's feasible and what can be accomplished now. And part of that would be, kind of like the efforts of training and community outreach in terms of where really the opportunities are in the use and applications of NAMs. But at the same time, acknowledging that there are limitations and there are still knowledge gaps that we need to fill in. And so be more realistic in how we create the not to overhype the technology, but to create sort of like a managed view of what the potential would be for this NAMS approach.
Yeah, just to add, agree with everything was said, but while we cannot control the narrative, and there are very powerful special interests from the animal rights groups and others pushing a certain narratives, I think we all need to embrace that and appreciate the challenge that is laid before us. And it's the job of politicians and and government officials to set ambitious deadlines. We also cannot argue with that. Where I think it's clear the rubber meets the road and the first real test of where the hype translates into actual uses is going to be confidence, and confidence can be built through painstaking testing of robustness and reproducibility. I don't want to use the V word, the validation part, because that has very specific connotations that are just impossible to apply to this particular field of science. But the work of establishing confidence is important, but it's also important to appreciate that no one really knows what the qualification looks like in this particular space, because the technology is both changing so rapidly, and also there are so many offerings. Going back to genomics, in reality there were three options, right? Whether it was printed, commercially printed microarray, your own printed microarray, or Affymetrix. There were three competing technologies, and then now there's RNA sequencing and a couple of other kind of smaller startups. In this particular space, the infinity is the answer to how many platforms are available, and testing and qualifying infinity is impossible. So European Commission is struggling with really what the qualification is going to mean. The OECD is struggling with how to adapt the validation framework to the new realities. And I think meetings like this and the communication that we have and the sharing of the ideas is really going to lead to that. But again, it goes back to, it's going to take us a decade to actually appreciate, or maybe for the market forces to work their way into going from infinity number of platform offerings to maybe less than 10. And then we can start thinking about proper validation and then proper regulatory qualification as well.
Okay. So next, a little bit of an advice question. What advice would you give to researchers working in the transplantation space who are currently using animal models to study recipient immune responses generated against an allogenic or xenogenic donor organ and evaluate novel interventions to prevent rejection or induce donor-specific tolerance? Are there any cost-effective platforms that could be used to make informed decisions about processes and targets involved in immune-mediated rejection of a transplant and or more complex biologics like CAR T cells? And some good questions today.
I mean, that's a complex question. there are a lot of immune competent in vitro systems. I guess that could fit this use case. I don't know. Dan, I saw you maybe unmuting. Maybe you have some in your toolbox.
Not quite. I mean, obviously these are miniature devices, so that it's not quite the proper avenue for testing large organ transplants, right? But you can test molecularly what's happening, as you indicated, Remy. There are a number of immune competent systems and platforms available for testing, whether it be devices or I've seen some, not entire organ systems, but for example, tissue patches that would be used for regenerative medicine and for tissue repair and wound healing. But not necessarily to evaluate an entire organ transplant system.
And yeah, just to add, I think separating the question, can this be done versus is this ready for being used in addressing specific questions? There's a big gap there. Innovation will not stop, and there are some really beautiful publications and and technologies that have been demonstrated to work in that particular lab. Now, I think it's important for the potential end users to identify the most promising technologies and then to start thinking about, again, testing their robustness and reproducibility. And having just in the last couple of weeks reviewing literature on secondary lymphoid tissue platforms in the context of vaccine candidate testing, I can tell you that the answer to other models is affirmatively yes. The question as to whether they're ready to be tested in a medium to high throughput in a BL3 environment, the answer is probably no. So again, this gap in, is the innovation there versus how usable that innovation is in a specific context of use is going to be the biggest challenge that we also have to address.
Thank you, Yvonne. Another question that seems to be for you as one of the speaker specific questions. What percentage of the models tested over the years are being used routinely by the consortium members?
So I think it's hard to put the percent on it, but I can tell you that the majority of the platforms that we're using, and there's actually not a huge diversity, there's probably less than 10 really commercial platforms that we have evaluated closely, or at least looked at in terms of feasibility of bringing them in and the cost associated with doing that. And what was surprising to me at the very beginning is that most of our consortium members are active users of MPS technology, and Roche and Remy's group is a great example. Roche has been one of the pioneers in actually bringing this technology in-house. And when they decided to join the consortium, I was stunned because, we have a lot to learn from them rather than to actually show them something. And equally true is for Genentech, who joined last year. They have a very large group that is actually innovating MPS at the company. But I think where the important distinction is trying to bring new platforms that really have not been tested quite a lot. And all of the commercial companies that were working with their models, they are innovating. So, they're releasing new platforms. And our consortium members want us to try first to see whether we can confirm the marketing materials that are produced by these companies. But also, we are testing different donors. We're testing different sources of cells. We're doing different type of phenotyping. We're benchmarking one MPS to the other. So we're asking very different questions. So I would say that majority of our consortium members have these platforms in-house, but they still participate and they renew their membership because we develop more detailed protocols. We test additional, we turn many more knobs that they can afford to turn in-house, and then they learn from that. And how they are using that platform in-house may be very different from how we tested it, but nonetheless, it's very useful still. So Remy, do you want to maybe put your perspective on that?
Yes, maybe one thing I want to say here is what's important to understand from our perspective is that we go by some sort of a motto saying, we use these tools are as simple as possible and as complex as needed. So we won't use like complex tools for the sake of complexity. We really want the simplest and easiest platform. And what I realized also seeing all this platform that Ivan lab tested in the past, often simple, 2D cell primary cells, let's say, or even cell lines do the job and quite well. So it's not perfect by any means, but if you add on top of the complexity, all the limitation of complex in vitro models, throughput, cost, familiarity, etc., it's difficult to show superiority to actually very simple systems. And this is a bit the reflection that we have internally, how we can manage this complexity and what is good enough. And often very simple models are good enough to answer our questions. Of course, we also explore much more complex system, very, very complex system, but the cost and the superiority in predicting human outcome is often questionable. That's maybe a point I wanted to make.
But also, just to briefly add, what we're doing in a consortium, not only benchmarking these complex models to more simple models, but we're also thinking about, some sort of a defined approach where you start with a simpler model as a funnel and then identify molecules that you really want to bring to a more complex system. And that's the only way to kind of go forward. So the question becomes not really, is this better than that, but what is the best workflow for us to actually have a confident answer? And the confident answer may include a very complicated model, but because of the cost and the low throughput of that model, it has to be put all the way to the end of the pipeline. And it doesn't mean that, intermediate complexity models, spheroids or transwells, cannot be used. So we have to be open-minded about what the NAM or MPS really is and not, trying to only advocate for very complex, expensive models. We have to look at the entire cycle of development and different contexts at different points of that cycle.
Thanks a lot. So for the next question, I'll try to combine two questions actually in one. So there is one for Dr. Teigel's talk that is about psychiatry. So it says psychiatry was mentioned in that talk, and this is difficult area for any model because brain functions emerges from interactions across large populations of neurons and multiple brain areas. So, and goes on explaining all that. So how are these issues being addressed in that area? And this can also be connected to another question also about researchers working on complex areas like aging and behavior that are whole animal, requiring whole animals. So would you like to tackle that, Dr. Teigel, and maybe the other speakers as well?
No, so certainly that's a very important point to bring up. Certainly that's a large area, even now, clinically and genetically, even though those are hard to figure out issues, right? And so having to, you can only model a system based on what you know. And so I think, but having said that, we have been, have had productive models developed from MPS, let's say even for pain, which is also equally something of a subjective nature, but we do know a lot about circuitry of pain, and so it's more of trying to understand what is the circuitry of a psychiatric disease, and how do those systems, the neural network, connect, and how are they disrupted. And so you cannot model something that you do not necessarily know a whole lot clinically about as well. We do know certain areas of the brain that might be affected in some of these conditions. But I think we are, as Ivan had said before, you can set the goals and develop the innovation and the technology that would address. But again, that depends on what the current knowledge is on a particular group of disorders, whether it be psychiatric disorders or behavioral disorders.
And related to that is the topic of what type of toxicities we want to model and replace. The reality is that most regulatory decisions are based on 28-day rodent study, 90-day rodent study, and the 2-year cancer bioassay. And the challenge of extending the longevity and functionality of these in vitro models, MPS or something else, has been the challenge that is very well recognized and people have been working on for decades. But I also wanted to, again, bring back as to what the goal is reduce, refine, replace animals. And to do that effectively, we need to understand where the animals are used. So European Economic Area is doing a great job releasing reports on where the animals have been used. And the largest number of animals that are used are in testing batches for, different vaccines or other biologics. And this is exactly where the neuromuscular junction and some other types of technologies can make a real impact in the total number of animals used. But for the chemical and drug safety testing, when you look at the both numbers and the types of animals are used, these are overwhelmingly rats and mice. And against the total number, the numbers are fairly small for chemical safety and for the drug safety use. And where they're used the most are these chronic assays. It's the developmental and reproductive toxicity multi-generation studies. It's the chronic carcinogenesis. It's the subchronic 28 and 90-day studies. And not only they're long, but also they're evaluating effects on any type of organ where things can go wrong. So remember, in the complex in vitro model, maybe we'll have different cell types, maybe we'll have different organs, but we'll not have 42 organs that the rat or a mouse has, and we really have no idea where things can go wrong. And that's why we're using animals. So again, we have to be realistic about what we can replace, what we cannot replace, but we can select areas that will be most impactful, and NCATS and their funding programs have been very effective in identifying those areas.
All right. Thank you for that. So kind of related to this is actually specific to the ToxBal Consortium. So Yvonne, this is for you. Where can labs go in order to kind of have an idea of what would be the best tissue to use or chip to use for their needs? Do they have to be members of the consortium? What about small academic groups that maybe can't afford to be members of the consortium? So what are the resources available for either CROs or small facilities to really make a decision as to what would be the right way to go forward, given the information that is available within the consortium?
It's a great question. And, to some people, $100,000 a year may look like a lot. To others, many of our consortium members, they probably will have to spend more to hire one scientist to actually do any research, let alone buy any of these expensive tools and gadgets. So, the cost set aside, what we're trying to do diligently is to share information. So, and the sharing of information is done not only through publications, but also through the EVE Analytics, now Curious AI database. So we release all the protocols, we release all the data, positive and negative, and we publish, and in those publications, we do make opinions as to what worked and what didn't work and what is quote unquote better or worse because we have relative comparisons. So that's why I included the list of publications. For those of you who are interested in particular organ system, you can go just find the ones that are on kidney, on liver, on on the heart, on the gut, and then start from there. Just see how we've designed experiments, how we conducted those experiments, and how we ask questions. And again, these are not publications in Nature or Science. This is not something that is super exciting, but this is super important work in my opinion, because this is the work that will actually build confidence in these models and will actually result in their use. So yes, our study designs are fairly straightforward and our phenotyping techniques are fairly straightforward, but that's what end users can only afford and realistically can implement in their labs. You heard Dr. Villanov saying that they have to be as simple as possible and as as complicated as needed. So please look at our publications, reach out to me. We'll try to answer any and all questions.
Thanks, Juan. Can you drop that link into, I think you can answer, there's one that says, can you post the database site here. Can you answer to that one?
Sure.
Or you can send it out. Yeah, thank you.
I mean, the chat is blocked right now, so yeah. So I think
it will be
it's
available on our webpage.
evanalytics.com. So EVEanalytics, one word.com. So
thank you.
Yeah, and I think unfortunately this was amazing. There was so many questions and so good discussions, and this is all things that are so relevant to all the work we are doing. So I really appreciate all your answers and how we can move forward and how we can, do improve even more and really get these kind of alternatives into use. So with that, I want to thank all the speakers again. Thank you so much for your thoughtful, sharing all your knowledge and also your perspective on this. I want to thank Dr. Renke and Dr. Mansouri for moderating the Q&A and also all the people behind the scenes. So Kathy Sprinkle, Stephen Moorfield, and doing the logistics, and the AV team, Nathan, Chris, and the captioner, of course, and all of you that have been listening and submitting all these good questions that we have been discussing. And I just want to highlight, I want you to come back. I want you to come back in 1 hour and listen into ICCVAM's public forum, as well as tomorrow. We also in the morning, we have our public forum, and we will actually hear more about the activities of ICCVAM agencies. So from FDA, for example, some of the things we have touched on today, as well as the NIH and NIHS, and we're even going to hear some of our own NAMS work on MPS on Tuesday, and some validation we have been doing for more complex models. But with that, thank you so much, and we're just on time, and I'll see you in an hour, I hope.